In their second paper on the structure of DNA, Watson and Crick described in general terms how the double-stranded structure of DNA suggests a pattern for copying the DNA during each round of cell replication. Prior to duplication, the hydrogen bonds are broken and the two chains unwind and separate. Each chain then acts as a template for the formation onto itself of a new companion chain, so that eventually we shall have two pairs of chains where we only had one before. This pattern has since come to be called semiconservative replication. During replication, the original double strand of DNA separates, and each strand is used as a template to create a new complementary strand. After one round of replication, this results in two double strands. Each contains one original and one newly synthesized strand of DNA. In subsequent rounds of replication, the same pattern persists. The two original strands remain intact, paired with newly synthesized strands after each round of replication. Today we know this is the pattern used by living cells, but the experimental evidence in support of semiconservative replication was not published until 1958. In the 5 years between Watson and Crick's suggestion and the definitive experiment, semiconservative replication was controversial and other patterns were considered. One hypothesized pattern was conservative replication. With this pattern, the original double-stranded DNA remained intact and was used as a template to create a completely new double-stranded molecule. A third hypothesized pattern was dispersive replication. This was similar to conservative replication in that the original double strand was used as a template without being separated, but prior to cell division, the strands recombined such that each daughter cell received a mix of new and old DNA. With dispersive replication, after each round of replication, the original DNA got cut up and dispersed evenly among all the new cells. From the perspective of today's understanding of molecular biology, the fact that this was a popular hypothesis seems odd. But the dispersive pattern was favored by several well-known prominent scientists. These scientists did not like the semiconservative pattern because they thought the rigid helical structure of the double-stranded DNA molecule would make it difficult for the strands to be unwound, separated, and copied in the way required for semiconservative replication to be possible. The dispersive hypothesis of cutting the helix every rotation eliminated the need for unwinding the helix. Proof of semiconservative replication was provided by a paper published by Meselson and Stahl in 1958. The methods they used allowed them to distinguish original DNA from newly synthesized DNA and to track the new and old DNA over several rounds of replication. They used different isotopes of nitrogen to distinguish existing DNA from newly synthesized DNA. To accomplish this, a culture of bacterial cells were grown for several generations in a media containing only N15, a stable, heavy isotope of nitrogen. After this period of growth, all of the DNA in the cells contained N15. These cells were then rinsed and put into a media containing only the more common, lighter isotope of nitrogen, N14. As the cells grew and divided in this fresh media, all of the newly synthesized DNA contained only the lighter nitrogen isotope, while the DNA from the original cells still contained N15. In this illustration, N15 labeled DNA is shown in orange and N14 labeled DNA in green. Meselson and Stahl then tracked the N15 and N14 labeled DNA using a high-speed centrifuge in a density gradient they created with cesium chloride. During centrifugation in a cesium chloride gradient, DNA accumulates in bands along the gradient based on its density. Since N15 is more dense than N14, N15 containing DNA accumulates lower down in the centrifuge tube than the N14 containing DNA. DNA with a mixture of N15 and N14 ends up in an intermediate position between the two extremes. By spinning DNA extracted at different times during the experiment, Meselson and Stahl were able to see how the new and old DNA was combined during each round of replication. The beauty of this experiment was that it allowed them to distinguish between the three different hypothesized replication patterns. At the beginning of the experiment, the cells contained only nitrogen-15. DNA extracted at this time and put into the cesium chloride gradient would only have one band low in the tube. If dispersive replication occurred, there would only be one band at all times, as the N15 labeled DNA was broken up and distributed evenly among all the daughter cells during each round of replication. After one round, all of the DNA would contain a 50-50 mixture of N15 and N14. In the next round of replication, the single band would become less dense as the original DNA continued to be diluted as it was evenly distributed among all of the new cells. If conservative replication occurred, after one round, the original band of dense N15 labeled DNA would remain and would be joined by a second, less dense band containing only N14. Additional rounds would result in more and more light DNA, but under ideal conditions, the original heavy N15 band would remain. With semiconservative replication, after one generation, all of the DNA is a 50-50 mixture of N15 and N14 labeled nucleotides, resulting in a single band halfway between the N14 and N15 only band positions. After an additional round, some cells would contain nothing but the light isotope, and some would still contain an equal mixture. There would be a band at the N14 position and one at the intermediate position. This is the key result. At the second generation, all three proposed replication patterns give different results. That Meselson and Stahl's experiment showed the pattern predicted by the semiconservative hypothesis provided the definitive experimental evidence in support of the pattern proposed by Watson and Crick. Here's part of the figure from the original paper showing changing DNA patterns in a cesium chloride gradient over time. In this image, the density gradient goes from low to high, from left to right. At time 0, there is a single, heavy, N15 only band. After one generation, the single band contains an equal amount of N15 and N14. After an additional generation, there are two bands, one in the intermediate position and a second at the less dense N14 only position.